Good afternoon, everyone, and thank you for being here. In attending the talks today and generally being in the neuroimaging community, I find that the mood of the moment is now to sit back and take stock of what we've done and how we've come this far. And in this regard, I'm going to introduce you to a new method and see what we can do further. So in the interest of reminding you about the variability of what we study, and to also just wake you up before the last session, I ask that you look at the screen carefully and then quickly press a button, air button, to respond what that monument was in the background. None of this should be surprising to you. You do this to your participants all the time. And not only that, you take pictures of the brain when they're lying in something like this, and then you stretch and expand and compress the brain into a space that doesn't really look like the brain anymore. Not so long ago, we actually used the space of a deceased elderly alcoholic French woman. And then we intend to find something about the brain. Great. Okay, so we've been doing this for 20 years, a lot, and we average about 15,000 functional imaging studies a year at this point. And it's a good thing we decided to take stock, because now for every 1,000 studies, we have a meta-analysis. And these meta-analyses are supposed to tell us, are we really finding what we intend to find? So what have they revealed? Well, most meta-analyses, like in most other areas of research, are topic-specific. And within topic, they find that, hey, more than one part of the brain seems to be working for a particular psychological experience. Now, you just saw with the variability in the questions we ask, the design of our experiments, the measures we take, the analysis we run, there should be no surprise that we all find different answers. How then do we reach a consensus whether we are actually looking at the truth? So thanks to some of our OHBM superstars right here, we have a lot of coordinate-based meta-analytic tools that are now available to us freely. With a click of a button, you get an answer to that question per topic. For example, with Neurosynth, you put in a term, and it scavenges the literature. It finds all the coordinates that are reported for studies that mention that term, and gives you, knowing the activation, the probability of whether that activation was related to the term that you looked for. Now, other than just giving you automated meta-analyses, which is great, these tools also give us a treasure trove of more data, like we needed it. What do we have? Well, from these databases, we now have topics or terms of interest, and we have voxels or regions of interest. Can we do more? It's a rhetorical question. Yes, we can. How? We first need to remember the nature of the data in these coordinate-based meta-analytic databases. They're binary. Was a voxel active or not? Yes, no. Was a term present in the abstract or not? Yes, no. Simple question, simple answer. So what do we want to know from these data? We want to know that in the past 20 years, throughout the literature, are there specific brain regions that seem to be reported specifically for some topics, and are there brain regions that are consistently reported across all topics of study? So we had a good starting point. My current PhD student, before he joined my lab, Fahad Al-Hazmi, was able to look at one half of this question and looked at the topics presented, actually the terms presented in the abstracts in Neurosynth. And he and his colleagues identified that even though at that point there were more than 10,000 studies in Neurosynth and using more than 3,000 terms, the variability in what people studied wasn't very much. There were actually only 6 stable clusters of topics that they identified that spanned the whole range of neuroimaging studies. And of these 6 topics, we were particularly interested in looking at 3 commonly examined topics: sensation and action, knowledge and language, decision and emotion. Specifically, we wanted to see for these topics, how do imagers report, or what brain coordinates do people report? So what was the brain doing? What do we have? We have a large database that needed some pre-processing. We actually have an asparagus versus pancake problem. I know I'm making you hungry before dinner, but believe me, there is a story here. So just to again remind you that we take images slice by slice, magically render it into this 3D space, call each little piece of this a voxel, then we string that voxel out into a vector, stack up these vectors like a pancake, and hope to analyze the data. Now the problem is, a lot of our statistical tools were developed 100 years ago, and they didn't deal with data like these. They instead dealt with data like this: more observations, fewer variables. We have fewer observations, more variables. I'm not telling you anything new again. We've kind of sort of solved that problem by using data reduction tools like the singular value decomposition and its cousin, the PCA. But there's a small catch here. These techniques deal with continuous data. We have counts. How many voxels were active for a particular topic? So if you want to look at the number of studies and the number of reported voxels, the solution is another cousin of PCA known as correspondence analysis, which is a technique that is specifically suited for counts and contingency tables, and has the added advantage that it's a bifactor model. That means you can look at the rows and the columns at the same time. So what does correspondence analysis do? For our analysis, let's start with the data in its rawest form. We had 141,000 voxels and 5,224 studies that we were interested in looking at from Neurosynth. When you have more information, use more information. So we know that we were not just interested in these studies on their own. We know that these studies were part of a cluster previously identified as being some of the stable clusters in the neurosynth literature. So neuroimaging literature. And therefore, we can classify the studies on the rows into the categories of interest. We also could classify the voxels into regions of interest to help better interpretability of the data. So with this data table, if I classify the rows and the columns into topics of interest and regions of interest, and collapse that large table into a table of sums or a contingency table, I am now reduced to a 3 by 63 matrix, where I can look at the 3 topics of interest at the same time as 63 regions of interest that included cortical and subcortical ROIs across the whole brain. The contingency table, so I analyze it with a correspondence analysis, and just like a principal component analysis, the results of a correspondence analysis are displayed in the form of orthogonal components that are extracted based on variance explained. These components show us the variability amongst the topics and the variability amongst the ROIs as reported for studies in these topics. For inference, to test the stability of these ROIs and topics in this component space, we use a bootstrap confidence interval procedure where we resample with replacement the topics with the studies within the topics and the voxels within the ROIs, and we generate confidence intervals around each of the original items. Confidence intervals interpreted just like always. If 2 intervals overlap, it means that these 2 items are not significantly different from each other. If they do not overlap, they are significantly different. So with our analysis, what did we find? We display component 1 on the x-axis and component 2 on the y-axis, reminding you that they are orthogonal. This becomes important when we try to interpret the results. In this data, component 1, which displays the maximum variability in the data, shows that there is the variability between the way brain regions are reported for studies on sensation and action versus studies and knowledge and language. That carries the maximum variability. Interestingly, component 2 that is completely orthogonal to what is described in component 1 shows that the way studies are report brain coordinates in for decision and emotion related work makes up component 2. It's not surprising that these studies are different from each other. They span different aspects of neuroscience. Now let's look at the brain and see what that tells us. If you notice, all 63 ROIs fall somewhere in the center of the plot. That means the whole brain is doing something at some point for each of these topics. But you will notice that instead of just being a blob in the center, there are some ROIs that tend to preferentially be reported for each of the topics. For instance, studies that examine knowledge and language tend to report more of the superior temporal gyrus. Studies that examine sensation and action tend to report more of the brainstem and postcentral gyrus. And studies that examine decision and emotion tend to report the amygdala and the accumbens, as compared to some of the other ROIs. What's interesting is to look at the number of ROIs that are still remaining at the center of the plot. What these ROIs tell us is, first, they are different from the ROIs on the edges, because the confidence intervals tells us they are, but also that these ROIs are consistently being reported, but they don't seem to have the specificity of the other ROIs. And we have to take a minute to remember that these are based on studies that actually report the ROIs. What then happened to the unreported ROIs? What do you think will happen to the plot? So a bit of an anticlimactic take home here. What are we trying to do? We are trying to bridge a gap, right, between what we are looking for and how we are looking for it. And we are doing it one ant at a time. So am I. And the idea here is that knowing the variability in fMRI and in neuroimaging in general, what is the best we can do with the tools we have? I've reintroduced correspondence analysis as an old technique. So reintroduce correspondence analysis in its new form that can examine the patterns in these meta-analytic databases and allow us to look at the studies and the voxels at the same time in order to find out whether we actually are finding what we think we're finding in all of our studies. We find that the participation of brain regions may have particular preference for particular functions, and that in getting a total understanding of the brain, while our individual studies could capture these specific functions, the variability and non-variability in imaging studies could just be a factor of what is being reported. And with that, I'd like to acknowledge all of my collaborators and my students and Neurosynth, of course, for the data. Some of this work is available on GitHub. And I leave you today with a modification of a very famous quote to kind of capture the mood of the moment. Thank you.
Thank you very much.